Okay, now on to hemoglobin physiology. Hemoglobin physiology is super important to know for the boards because of how it carries oxygen and the normal physiologic and the pathologic stuff that can happen to it in terms of hemoglobinopathies and thalassemias. Adult hemoglobin is a polypeptide comprised of 2 alpha and 2 beta subunits, with each subunit containing a non-heme protein group with a single iron atom that forms a strong covalent bond to a histidine residue in the globin chain. There is a taut form, which has a low affinity for oxygen on the heme molecule, and the relaxed form with almost 300 times greater affinity for oxygen. Now, does carbon dioxide bind to heme? No, it binds to other parts of the hemoglobin protein to form carbaminohemoglobin. Carbon monoxide, on the other hand, does competitively bind to the same heme site to form carboxyhemoglobin, which we will discuss in the next fact. An important concept to understand is the positive cooperation that hemoglobin demonstrates. So what's positive cooperativity? So what happens here is that the binding of the first oxygen molecule pushes the hemoglobin into the R, or relaxed state, by tugging on the histidine residue. So here we're going from taut to relaxed, and it becomes easier for oxygen to bind to other heme groups. And again, I'll repeat, taut, which tends to be deoxygenated, and relax, which tends to be oxygenated. This explains the sigmoidal curve to the oxygen hemoglobin dissociation curve, which we'll discuss shortly. Negative allostery occurs when something like 2,3-biphosphoglycerate binds to hemoglobin and stabilizes the taut form, as you see here, and reduces its affinity for oxygen. So how is this different from myoglobin? Well, myoglobin is a monomeric heme protein similar to the alpha subunit of hemoglobin. And because it's monomeric, it's not capable of having these quaternary structural shifts mediated by allosteric interactions. Finally, a quick word about fetal hemoglobin. Fetal hemoglobin is composed of 2 alpha and 2 gamma subunits rather than beta, which you see in adult hemoglobin. And this has a higher affinity for oxygen than adult hemoglobin. So here is the classic hemoglobin dissociation curve. And here you can see the difference in the oxygen saturation curves where the fetal hemoglobin curve is left shifted. So what do you think this is? Well, it makes sense because fetal blood has to acquire oxygen from maternal blood, so it has to bind with greater affinity. As you can see here in this diagram, fetal hemoglobin pretty much dominates from week 10 till birth when it rapidly tapers off after 6 months. Alright, let's talk about a couple of highly testable conditions that can modify the effectiveness of hemoglobin, leading to tissue hypoxia from decreased oxygen saturation and content. Methemoglobin is the oxidized form of hemoglobin containing ferric, Fe3+, rather than the ferrous, Fe2+ iron. This form of hemoglobin is less effective at binding oxygen and has, interestingly, an increased affinity for the cyanide ion, which we'll talk about in a bit. So how do you get methemoglobinemia? Well, actually you can get it from a couple different ways. Methemoglobinemia can be caused by inherited problems from cytochrome B5 reductase, or methemoglobin reductase, which converts methemoglobin to hemoglobin. This is an autosomal recessive condition that sometimes shows up in inbred families. In hemoglobin M disease, the defect is in the hemoglobin itself. However, more commonly, the condition is acquired via exposure to oxidizing medications such as benzocaine, dapsone, and nitrates. Methemoglobinemia can be treated with methylene blue and oxygen supplementation. However, methemoglobin itself is actually useful for cyanide poisoning. So how does that work? As I mentioned here, methemoglobin preferentially binds cyanide over oxygen. So the patient is given nitrates to convert hemoglobin to methemoglobin in order to bind the cyanide. Thiosulfate is then used to bind this complex cyanide, forming the thiocyanate, which is then renally excreted. Carboxyhemoglobin is typically caused by carbon monoxide poisoning, where hemoglobin binds to carbon monoxide rather than to oxygen. In fact, carbon monoxide has almost 200 times greater affinity for hemoglobin than does oxygen. Carbon monoxide binding means that there is less hemoglobin available for oxygen transport, as you can see in this graph. On top of this, there is a left shift, you can see over here slightly, in the oxygen hemoglobin dissociation curve, which means less oxygen offloading in the tissues. So how can you tell the difference between someone with methemoglobinemia and carbon monoxide poisoning? Methemoglobin makes the patient cyanotic. On the other hand, carboxyhemoglobin makes the blood bright red. Thus, you might have heard the mnemonic, when you're cherry red, you're dead. So finally, how do you treat carbon monoxide poisoning? It can be fatal, but you can treat carbon monoxide poisoning with high flow or hyperbaric oxygen treatment. The oxygen hemoglobin dissociation curve is another super high yield concept that you will almost be guaranteed to see on the boards. So you may recall from our discussion of hemoglobin that the binding and unloading of oxygen from the hemoglobin moiety shows cooperativity, which explains the sigmoidal shape of the dissociation curve. As we mentioned earlier, myoglobin as a monomeric globin protein has a sigmoidal shape, where it has a greater affinity for oxygen than hemoglobin. Interestingly, the hemoglobin curve can shift to the left or to the right under specific conditions. So why would the curve shift, and why would you care in the first place, other than to pass your boards? A shift of the curve to the right signifies a decreased affinity of hemoglobin for oxygen. What this means is that there is more unloading of oxygen to tissues. Think for a moment about situations where this would be helpful. Alright, for example, when you're exercising, your muscles need more oxygen. So it makes sense that the curve shifts to the right, meaning hemoglobin at the same PO2 will be less saturated since oxygen is being offloaded in the tissue. So there are a number of factors that will shift the curve to the right. The mnemonic to keep in mind is CBEAT, where you have CO2, 2,3-biphosphoglycerate, exercise, altitude, acid, and temperature. If you think about it, what do all these factors have in common? They all signal a need for more oxygen delivery to the tissues, and they all stabilize the T form, TOT form, you remember, of hemoglobin over the R relaxed form, which lets hemoglobin more easily unload the oxygen in the tissues. So this makes sense, right? Active muscles, like during exercise, locally cause an increase in temperature of the tissue, increase CO2 locally, and increase acidity. 2,3-biphosphoglycerate is synthesized in red blood cells from 1,3-biphosphoglycerate. Oh, wait a minute. 1,3-biphosphoglycerate sounds familiar. Where have you heard of this? 1,3-biphosphoglycerate is an intermediate in glycolysis. Making 2,3-biphosphoglycerate actually costs the red cell 1 ATP that it would have gained from converting 1,3-biphosphoglycerate to 3-phosphoglycerate. But in return, the body gets a molecule that is critical in shifting the dissociation curve when it needs more oxygen. So when does the body make more 2,3-biphosphoglycerate? You usually see more of it when there is tissue hypoxia, like at high altitudes, in COPD, which we'll discuss in the next talk, and CHF. Again, no shocker here, these are conditions that deprive tissue of oxygen. Not surprisingly, if you decrease these factors, you can get a left shift of the curve, which means increased affinity of hemoglobin for oxygen, thus less unloading of oxygen to the tissues. As we discussed earlier, fetal hemoglobin, or HbF, also shifts the curve to the left because it has a higher affinity for oxygen than adult hemoglobin. And this is mainly because it has low affinity for 2,3-biphosphoglycerate, so at baseline, the dissociation curve is shifted to the left. Finally, as we discussed earlier, carbon monoxide shifts the hemoglobin curve to the left, but ultimately limits its carrying capacity.